Precision medicine has really kind of revolutionized where we are now in terms of bringing personalized therapies to patients. So we've seen a variety of new treatments come out, largely out of what we call basket trials, which are looking at patients with a specific genetic marker, but now across all tumor histologies or all tumor subtypes. What they're looking at is now they can look at, well, do we have an inhibitor for mutation X or mutation Y? And can that have an effect across, again, multiple different disease sites?
The real power of precision medicine and the sort of hope offered by precision modeling of individual patient tumors is accentuated by this whole idea that we can really precisely treat the disease the person has and give them the right care for the right patient, which is a really exciting place to be, especially as a scientist who is, has a library of drugs to play with.
Precision medicine is also called personalized medicine. It targets the right treatments for the right patients at the right time. It is a new type of biomedicine that allows you to understand the body at a molecular level. Therefore, the goals for precision medicine are in cancer research, molecular diagnostics, CRISPR, and immunotherapy.
The recognition of the FDA and the EMA actually to reduce, number one, animal testing. So they enable to use ex vivo models to do this. There are like organoid chips, obviously. There are patient-derived organoids as well in this space. Some research can be done on iPS cells that are embedded in 3D matrices. And so I think just that recognition that the highest agencies for those approvals are recognizing those models, this is kind of key for the field to move further and to actually now make the impact and close the gap from the laboratory setting to the bedside and to really bridge that translational relevancy.
One of the big problems of precision medicine is it's very expensive. I think with organoids, so together with some companies, we have indeed been able to move some of the compounds that were developed on organoids already to a clinical phase. And we think, indeed, and hope that that actually will have a major impact, not only to make better drugs, but also to make more affordable drugs.
Some of the things I think are the most exciting are having patients' genetics, their environment, them in a petri dish, patient avatars, and then also being able to do things and test drugs that we wouldn't want to do in a human just yet. We can knock out any gene in a petri dish, you can't knock out any gene in a human, and so really having these tools to dig at mechanisms before we move to the clinic or go any further.
Where we need to go and where some groups are already starting to explore is how do you improve the quality of life for these patients in terms of side effects from drugs? So can we use organoid models to predict which particular patients might suffer from a particular side effect and kind of head that off from the beginning?
Organoids, or the organoids that we at Hub Organoids use, are adult stem cell derived cultures. That means that we can take a biopsy or a sample from a patient and take that to the lab, and we grow it out into organoid cultures, which we can then put in the freezer and we can use as a platform for research and drug development. Organoids, of course, as a new technology, are in itself quite expensive. On the other side, the drug costs, there we have to see how it works. I think it is an expansion of using organoids. We want to bridge to the clinic, stratify patients easier so the clinical trial becomes cheaper. And so all these steps, we're really early on. We really think that will have an impact in the coming years.
A lot of the challenges we're seeing in precision medicine is reproducibility and scale. So how do we make a good model for a patient and then benchmark it and then translate that to other patients? So what are the readouts that's actually predictive of that patient's response? How do we look at a patient's organoid or a model organ in the dish and say it's responding like the patient would? And then if we do get that, how do we do that for every single patient or bulks of populations at scale?
For our center in particular, we're limited in terms of the availability of tissue. So for small cell lung cancer, many of those patients do not receive a surgical resection when they present with disease. They go straight into standard of care chemotherapy. So we're generating these tumors kind of from scratch by taking established cell lines in the SCLC community, putting them into our organoid models, and then creating these miniature tumors from there. So that's a big challenge.
In the next 10 years, we're starting to develop these umbrella trial protocols and the ability to specifically target individual tumors. But the real need is to show that our modeling approaches are really able to say, this drug is the right drug for this patient, and thereby build out our ability to take the data that we're able to generate now at high throughput for individual models and apply it to individual patients. And so that's really the key barrier that I think correlative studies and the ability to do co-clinical trials will expand and allow us to sort of step into in the next 10 years. My lab's work uses patient-derived organoid models of individual tumors. And so what we do is to take primary tumor samples from the clinic, dissociate them, and then grow them in 3D matrix using Matrigel to create tumor avatars for every patient. And we do that in high throughput and replicate so that we can create hundreds of thousands of copies of the same tumor to run high throughput drug screens.
So for modeling human disease, we take an organ on a chip approach, but it's a combination of organoids or organs on a chip. So when you're doing organs on a chip, you'll define a function of an organ. So something you can measure. So for the heart, it's contractility, so it's beating with force. That's something we can measure, and we'll focus on just making a tissue that has force in a petri dish and what we can measure of that.
At Cold Spring Harbor, we use patient-derived organoids that were grown in pure Matrigel in domes, and we would use those to predict cancer therapies for patients. The other approach that I've now followed up at the company that's called Xilis is they have developed something that is called a microorganosphere, which is an encapsulated cells in small Matrigel droplets so that you retain in a very small environment the cells that are coming from your biopsy or your resected tissue. And this can be now propagated. And this is kind of, I think, one of the key differences to many other companies and approaches that I've seen so far.
The models that we use are kind of twofold. We have normal lung organoids that we make, and we also make normal cerebral or brain organoids. And that's because we want to model the primary tumor. So for small cell lung cancer, it grows in the lung, right? And one of the main organs that it metastasizes to is the brain. So we like to have liver and brain and compare how these tumor cells grow within these different microenvironments and see how they have different responses to different treatments and therapeutic interventions.
We love patient and mouse organoid models. And so my colleague Josh Smith and I, who's a surgeon scientist and a colorectal surgeon, he's helped develop the Memorial Sloan Kettering Biobank for rectal tumor organoids. And we've developed matched tumor and normal organoids so we can assess relative radiosensitization on a patient-specific basis. We also have developed immunocompetent mouse models to study the interaction of radiation, also chemotherapy and targeted therapies with the immune system. So we can really kind of recapture and recapitulate the effect of what's happening not only in a tumor intrinsic manner, but a tumor extrinsic manner, capturing those immune effects.
So at Hub, I think most of the work consists of, on request of industry generally, to make specific models to ask specific disease questions. For instance, in inflammatory bowel disease, we need to study barrier function, so we make models in which we can actually measure barrier function, disruption or repair when we're testing the drugs. So making assays is probably one of the biggest things, and also where all the developments are by adding immune system, adding fibroblasts also, and to further develop these initial organoid models.
Corning Life Sciences is a leader in ECMs that are needed to grow organoids, such as the gold standard Matrigel matrix, collagen, and laminins, as well as Corning Transwell permeable supports. In the scaffold-free space, we offer spheroid and alplasia platforms, clearing reagents for better imaging of 3D structures, organoid counting cell counter, and our newest launch, the Corning Matribot bioprinter. Therefore, our approach is to stay closely connected to our customers so we can provide these tools in a timely manner to enhance precision medicine.
So there's always room for improvement to our models, and some things that are really exciting for us are putting in all the cells that are in each environment. And so currently you'll see the main cell type in each organoid or tissue model, and maybe one supporting cell, but really having immune cells, all your stromal components, vasculature, having everything, all the right cells at least there. is the next building block.
We and other groups hope to incorporate other cell types within the organoid model. So for instance, in the brain, we see neurons, we see glial cells, we see important structures like the choroid plexus, which produces cerebrospinal fluid in the brain, but we don't have endothelial cells, we don't have immune cells. And so, whether we can incorporate those through differentiating our starting material into those cells or co-culturing those types of cells with our organoids, we hope to make it more of an all-inclusive model to be more representative of a patient organ.
In the models we're currently using, there's certainly opportunities to improve them. One, right now, even though we have this really large biorepository of patient-derived tumoroids, really our biggest challenge right now is trying to find the ability to screen and assess and characterize them most appropriately.
So there are still protocols missing with growth factors for certain cancer types. People have seen how your current media formulations can actually change and put pressure on the state of those cancer cells. And I think figuring out in the next years what those effects are is gonna be key to establish more of those relevant models.
We're better able now to build models with T cells and tumor cells and complex interplay, but trying to scale that and really optimize it for each individual patient in a way that is clinically relevant and can be used rapidly, right, is a major limitation that obviously we're going to have to address.
This assay technology can use improvements in standardization, reproducibility, and automation, which are all necessary to fulfill the translational potential of patient-derived organoids.
What we think is the ideal use of organoids is this patient stratification. So it's basically to really use its characteristics as a patient-like sample that you have in the lab to look at a bunch of patients, at a group of patients, and predict how that group will respond when you move your drugs forward in a clinical trial. So from our perspective, something that we can already do, but is a conceptual change on how we do drug development, is really to start using it as this patient in the lab, seeing it as a single patient or as a group of patients, and use the organoid as a platform like this, more than as a traditional sort of fancy HeLa cell.
We haven't, had a chance to look at this in animal models before. So cell therapy, CAR T, all your T-cell antibodies, the whole future of medicine is very alive per se. And so these models let you screen that really, really well.
One of the exciting things that we see in our models is that we see that the chemotherapy sensitivity and radiation sensitivity of our human patient organoid models, it really correlates or associates with what we see in the patient. Patients who have sensitive organoids that respond to the chemotherapy, they live longer without having disease recurrence. So that's a big kind of thing that gets us excited that these may help us kind of infer or predict what's going to happen on a treatment basis level, where we can test new drugs, new therapies, new strategies to bring into our clinic.
Now you can grow ideally a patient in a dish and can experiment on this patient, expand this biomass indefinitely, do your proteomics, do whatever method is developed in the next 3 years, and you can always go back. You are not losing any of this material.
Biobanking tumor organoids from patients is a very large enterprise. It takes essentially an army of individuals from consenting patients, collecting specimens, transporting specimens, growing them in the lab, changing them, establishing them, characterizing them. And so what we find is that it actually takes a large amount of kind of support. Are we just going to focus on one disease site? Or are we going to focus on patients on clinical trials, or every patient that walks in the door? What's the scientific question? What's the clinical question? And making sure that we are kind of assessing and tweaking our biobanking needs accordingly to meet those goals.
So on the one hand, we have this the patient itself, so getting all the samples in. It's working out that we need more, and we need to get these arrangement hospitals in. And sometimes also explaining to patients, why do we want them, why is it useful? On the other hand, the reagents are an important part. So the Corning and Stem Cell Tech and some of these companies have developed an enormous amount of reagents by now, which is very important because they help us to standardize it. So it's basically the characterization, standardization, and resourcing that are under development and are moving forward, but there's still a lot to do there.
lessons I've learned in biobanking is mostly it's hard to make models reproducibly and reliably, and then once you have it going, things change that you don't expect. So COVID could shut down your lab and you have to freeze everything down, or you have changes in staff and handling's different. Patient samples come in, they can be very different. And so it's just getting the workflow very standardized to help biobanking and make it reproducible is somewhat challenging.
Having the ability to say, we have developed organoids from 400 patients and we think that this subset is going to be sensitive to this specific approach. You can then go back to those specific 6 tumors, let's say, and really focus and personalize their care in a way that you could never do if you didn't have a biobank. And so the preservation of samples, whether it be for genomics later or functional modeling later, is a critical component to being able to really build out the promise of precision medicine.
High throughput drug screening is where we want to go with our current model system. We want to not only screen the lung and brain SCLC organoid tumors with standard of care like etoposide and platinum-based therapies, but we want to also look at other new therapies, novel therapies that maybe no one has thought of using in the clinic, and see if certain molecular subtypes of this tumor respond well to a certain treatment versus others, and really be able to take this down that route of personalized medicine for the patients.
When people are talking about all these models, we sort of take for granted the fact that we all are plating in this 3D matrix that provides us with a physiologic feedback. When you do a 2D patient-derived line versus a 3D patient-derived line, the number of drugs that the 2D line responds to versus the 3D responds to are very different. And then there are a couple where you will have an inverse where in general the organoid is less sensitive and the 2D line is more sensitive to everything, but there'll be a couple where they're switched because the matrix is creating a vulnerability. And the better the models, the more we become dependent on the structures that hold them together, right? So if you're doing a complex skin model versus just a simple tumor organoid, those types of matrix components are fundamentally important.
As interest in organoids is expanding and more researchers are starting to utilize them, both from mouse and human, what we're finding is that now many companies are developing products specifically geared to organoids. Whether this is something like having tailor-made medias, which makes it much easier and accessible to a wide audience, or whether having new microscopes that can either live in incubators or that are specifically designed for organoids, that can monitor organoids in real time. Those are really kind of things that are now helping improve our ability to assess and do screening-based methods.
The aspect, I think, to improve the drug screening with organoids, we're developing special plates that really fit your needs for the drug screen itself. In my case, this was a 1536 row plate that needed to be developed. Beside this, I think liquid handling is one important step. Obviously, having high humidity incubators is key. And I think a robust setup for your automation is just important here.
Significant impacts in screening, mostly all the automation. So everything's, even your drug dispensing, your plate reading, everything's automated. And then organoids, Matrigel, organoids, the whole system works really reliably. You can have patient cells with ease. These are things we couldn't do years ago. And so it's really changed the field. And it's exciting to not always just rely on cell lines, to have 3D models with a better tumor microenvironment so we can really see more reliable results.
So there have been quite a lot of tools now that are developing, both in equipment but also in plates. The plates are now more organoid specific. When we made the first organoid, there was no specific organoid equipment, of course, because we were the first one making them. And actually that was a problem because a simple example is ultra-low attachment plates that were rarely used back then, but were key to organoid research. Since 2009, there has been an enormous increase in the number of resources that have been generated by the supporting companies that we now use in screening regularly, but were not existing 10 years ago. And hopefully we'll have a lot more in the coming years.
We talked about cost as a factor. Decreasing costs, for example, in genome sequencing comes to mind. Development of automated cell processing systems to generate highly pure, reproducible and consistent population of cells. Improvements in computational technology is another item that needs improvement.
Whereas we have developed these drugs very specifically in the preclinical phase, we have very few tools to allow us to actually screen which patient could benefit from this precision medicine. So this precision medicine is not very precise at all. And there I think we are going to see a big difference, because now we have models that are specific to patient, and we have the drug developers, the chemistry, to make the compounds that are required for it. So I think indeed now we will really be starting to work on precision medicine.
There's a lot of unmet needs when we're thinking about kind of where the organoid research is going and how it's being utilized. A few of them, include really developing new technologies to allow us to kind of rapidly screen organoid sensitization and resistant mechanisms. Others include looking at really kind of how's the heterogeneity of the organoid cultures changing and how is it evolving and adapting.
We hear a lot about using organoids and 3D models for drug screens, and that's what we're interested in doing also. But as they say, the proof is in the pudding. So being able to show that your preclinical screens in organoids actually translate into true efficacy in the clinic would be amazing. And I think that's where we really need to go in terms of translational medicine and improving precision medicine.
To get prescribed a drug, you need to show that it'll have a benefit to the patient. And so a lot of times you'll wait until a very late stage in the patient's course of treatment to give them a drug as like a last ditch hope. But instead, if you have patient organoids, that could be your biomarker. So you can prove from the get-go, this patient will respond, should respond, and then get approval to get the drug.
While it's great that we can develop these huge banks and run all these screens, the key to going from using that to inform a targeted clinical trial to actually informing patient care is something that is sort of waiting. And the only times that we use it now are in areas of compassionate use, or when there's an FDA approved drug that an insurance company is willing to pay for, because a tumor has a specific mutation that it shouldn't. BRCA mutation in a pancreatic cancer case. The ability to scale and sort of correlate our actual responses is really the key missing agent.
And a new application of precision medicine is where we think we're going to see some major differences. And to explain it, use the example of rare diseases. Cystic fibrosis is a disease that we study at HUB and work with the industry for at HUB a lot. And the interesting part there is that it has a wide range of genetic variations, of which many are untreated at the moment. What we can do with organoids is that we basically take a sample of the patient with rare mutations for which there's no treatment. The sample we translate to organoids, we bring it to the preclinical side, we bring it to our industry partners, and test the drugs that they're already making or that they're trying to make. And we find drugs that are specific for this patient group that indeed has a very high unmet need. And then translate this to clinical trial and basically back to the patient. And that circle is actually something that works. We are already doing that. So we have a collaboration with Xilis, for instance, that is developing a drug for a specific group of patients that was not treatable before. We make organoids of a potential group, we test them, and those patients go into clinical trial and then are treated with this new drug.
For future applications in precision medicine, I think the goal should be to have a biomarker-based approach that can be even taken by people at home, even before they are getting sick or at their annual physical exam with the doctor, so that everything gets as easy as just taking a pill.
Patients who suffer spinal injury, I saw a really interesting paper not long ago where they were able to sever the spinal cord in a mouse, take a cerebral organoid, put that into the spine, and then watch the neuronal signaling reoccur. And that's amazing to me. And so I feel like outside of cancer, there are so many applications for organoids and 3D modeling to help many different types of diseases.
Imagine you need a knee transplant. Stem cells are harvested, bioprinted in the shape of a knee, and then transplanted back into your body. That seems to be the pinnacle of precision medicine for me. Another interesting area is the combinatorial approach of using organoids and immune cells to study infectious diseases. In fact, organoids were used to understand COVID-19 and Zika virus mechanisms, and it showed that they have impact on the GI tract and brain respectively. Therefore, the combination of organoids in vaccine development is very relevant in these times.
When we first made the organoids, we thought we had a very interesting scientific tool. For the first time we had genetically stable cells in the dish. And we were hoping for a lot of things that were predictive for patients, etc. So the surprising thing that happened to me actually was probably that it actually was true. So initially we were hoping a lot of things when Toshio Sato made the first organoid. And now in these meetings 15 years later, all of these things are actually coming true, much more in that sense than we might have expected. So indeed, those initial organoids do seem to be a patient-specific tool that we can use in all these different applications.
One time I was doing a screen and I saw all these immune cells just come into one of the tissues. Well, I've never seen this before, what's going on? And it turns out I had a contamination, which was not a good thing, but it helped us to see that our immune cells really trafficked into organs as they should. And so now we do injuries where we can actually elicit that immune response. It's not a contamination, but it's, you'll see the immune cells come in, they'll go right to the site of injury, do their repair mechanism, and we can do it with drugs as well that elicit the immune response. But it was really nice to see that if we are vascularizing all our organs and having this circulation, that you're still seeing what you see in the body, whereas if you have an injury or even if you have an infection, contamination, the immune cells just go right there and take care of it. And it's just exciting to see in a Petri dish.
When you give a targeted inhibitor, one of the things that happens at the level of the transcriptome is that pathways go the opposite direction. You get lots and lots of transcripts for a pathway that has been shut down. And so then unraveling those types of what is actually happening in the tumor can be hugely beneficial for then identifying which of the drugs do we want to pick for it. So if you've got a tumor that is dysregulating purine biosynthesis and you have a purine analog that it's synergizing, it may not be the best hit in your screen, but you have a molecular basis by which to say this is how this is working. And those types of surprises where you're just this is backwards, I don't understand, and you have to sort of bang your head against the wall for a bit are more and more common the more you look at individual tumors.
I was really surprised about how quickly the cells would invade. So previously I worked with glioblastoma rather than small cell lung cancer. And so glioblastoma or GBM is incredibly invasive, but working with small cell lung cancer, they just attack immediately and invade into these organoids within a matter of hours. So, it doesn't even have to be 24 or 12 hours, it can be less. So I think that just speaks to how quickly they can invade in a patient's organ and why, there's such a significant unmet need for this patient population.
I remember the first patient we'd derived organoids from. It was a young man with cholangiocarcinoma, widely metastatic. He said, hey, I want to donate my cells to you to grow organoids. And so we were fortunate to get these and grow them from him. And when we put them into mice, what was really interesting is that the organoids had a tropism. They would go to the exact spot where we isolated them from and grow very nicely, but they wouldn't grow in other parts of the body. And even if you injected them in the vein or in the belly or by any different methodology, what you'd actually find is that some way they would find their way back to the niche that they liked and grow. So it's always been something really fascinating to me, this kind of memory that these organoids have, especially not only memory from a human to a mouse, that they retain that. It's something that always strikes me in terms of, just how special these organoid cultures are and just how much we have still to learn from them.